guys, it's Mrs. Guido. I just wanted to go through the review guide with you. This test is going to be over meiosis and protein synthesis. So the first part of the review guide was a refresher on meiosis. This is the first page. So it says below are the steps of meiosis. Out of order, label each step. These are the same steps that you guys got before spring break, where we did like the cutting and gluing. So you cut these strips out and they were out of order. You had to identify each of the steps and then place them in order. You got the stamp and then you drew each step next to them. So this is like a multi-day activity. So let's go through this. The first one says DNA replication happens before meiosis. So this technically isn't a part of meiosis, but it is an important step. And remember that DNA replication happens during S phase of interphase. Oopsies. So we'll say that this is interphase. You could say S phase. Either one works. Let's on the side write down the phases of meiosis. So we have meiosis, and meiosis is made up of two steps. So we have meiosis 1 and meiosis 2. So meiosis 1, remember it's PMAT. So we have prophase 1, metaphase 1, anaphase 1, and telophase 1. Then we go straight into the second phase of meiosis and meiosis 2, and that goes into prophase 2, metaphase 2, anaphase 2, and telophase. Oops, why did I do a P? My bad. Telophase 2. And remember the telophases are like slash cytokinesis. So cytokinesis is also happening, splitting of the cytoplasm. And then that's the end of meiosis. So meiosis is made up of, again, two phases, meiosis 1 and meiosis 2. So now we need to make sure we have all of these orders together. So it says tetrad lines up in the center of the cell. They are attached to spindle fibers. So we have some key words that are happening. So we have the tetrad lines up in the center. So if it's happening in the center, we know that's going to be dealing with middle. If the tetrad is there, then that means that we must be in meiosis 1, because in meiosis 2, the tetrad has separated and now they're in two different cells. So this is going to be metaphase 1. Cool. The nuclear membrane forms. So that's a key giveaway. I know that when I see that, the nuclear membrane forming is meaning that I'm dealing with a telophase. Let me do this actually in a different color. Will that go over it? No, that's all right. So we're going to be dealing with telophase. So you have to figure out, are we dealing with telophase 1? Or are we dealing with telophase 2? So the nuclear membrane forms around two groups of chromosomes. Well, if we only have two groups of chromosomes, then that means that we are going to be in telophase 1, because we start off with one cell, we end up with two cells. And then those two cells go through metaphase or meiosis 2, and then they split into our 4 cells. So 1, 2, 3, 4 when we get into telophase. Okay, so this is telophase. Telophase 1. All right, the next one, sister chromatids separate. So let's get a different color. So sister chromatids are separating and move to opposite ends of the cells. So when I have things separating, I know I'm dealing with anaphase. Oh my gosh, that looks awful. Hold on. We'll just circle it. Anaphase or anaphase 2. So you have to figure out, okay, are we talking about anaphase 1 or anaphase 2? When we're looking at these in anaphase 1, this is where we have our homologous chromosomes. So the homologous chromosomes are in anaphase 1. In anaphase 2, this is where the sister chromatids are. So anaphase 1, the homologous chromosomes separate, and in anaphase 2, the sister chromatids separate. You do need to know the difference between those two. So since this is saying that the sister chromatids are separating, we're dealing with anaphase 2. next one, produces 4 haploid cells. So it means we are resulting in 4 haploid cells, produces 4 haploid daughter cells. Each has half the number of chromosomes as the parent. So if we are producing 4 haploid cells, well, over here we said this is where we make our 4 cells. So that's going to be our telophase 2. Okay, the spindle fibers pull apart the homologous chromosomes. We just talked about this. So the homologous chromosomes are the one that we just said right here, and that was in anaphase 1. Remember, the homologous, homo means same, so these are chromosomes that are coding for the same genetic information, and that's where crossing over can happen. So when homologous chromosomes pair up and form the tetrad, crossing over can happen, and then after crossing over happens, they line up in the middle, and then they can separate. Okay, the next one. 2 daughter cells are produced. So 2 daughter cells are produced, each having chromosomes that are different from each other. And I think that actually in this one we did have meiosis 1 section and a meiosis 2 section. So I might have to go back and edit some of these. So 2 daughter cells are produced, each having chromosomes that are different. So that would be the end of meiosis 1. Okay, centrioles separate and produce spindle fibers. Nuclear envelope breaks down, chromatin condenses into chromosomes. So this is going to be either prophase 1 or prophase 2. That's what we're looking at. And remember in prophase 1, this is when things get a little bit wild when we have our tetrads forming. And this has nothing to do with the tetrads. It's just kind of like what has happening in mitosis. So that's going to be our prophase 2. Okay, the centrioles line up in the center of the cell. It does not say, the chromosomes line up in the center of the cell. It does not say homologous chromosomes or tetrad, so I'm going to assume that we are looking at metaphase 2. Okay, the nuclear envelope forms around the 4 groups of chromosomes, So that's actually going to be our telophase 2. And you might be like, wait, we already used telophase 2. I know, my bad. Hold on. Up here, we're going to erase this one right here. Instead of this being telophase 2, what would it be? It's either going to be meiosis 1 or meiosis 2, and this will be meiosis 2, the end result of meiosis. Oh my gosh, can't spell. Here we go. Meiosis 2. Okay, so I hope that makes sense. So when we're looking at, we have our meiosis 2, and then we had our meiosis 1. They're just showing our end result. So we produced 4 daughter cells, and then at the end of meiosis 2, we produced 2 daughter cells at the end of meiosis. The end of meiosis 1, we produced 2 daughter cells. The end of meiosis 2, we produced 4 daughter cells. And then the other ones are just going to be the steps. Okay, each chromosome pairs with its corresponding homologous chromosomes to form a tetrad. Okay, so we have our homologous chromosomes pairing up to form that tetrad, and we have crossing over happen. So whenever you see crossing over, you should be thinking of prophase 1. So that is our prophase 1. And I think that we have one more. Centrioles start moving, spindle fibers form, nuclear envelope breaks down, and chromosomes uncoil. So what are we missing? Centrioles start moving, spindle fibers form, nuclear envelope breaks down. So we have PMAT, PMAT, and PMAT. You know what I think I did? I added more to this for prophase 2. So you can actually just ignore this one. Just ignore it because we already did prophase 2. I think I just wanted to have like all the things happening in it. So like centrioles producing spindle fibers, breaks down, chromatin condenses into chromosomes. Yeah, okay. All right, so the purpose of meiosis. So why do we go through it? Like, what's the point? The purpose of meiosis is to produce 4 genetically different haploid cells. And remember, haploid means half the number. So in humans, if we have 46 chromosomes, when we go through meiosis, we end up with 2 cells that now have 23 chromosomes. Sorry, we end up with 4 cells, my bad. And each ends up with 23 chromosomes, and they all look genetically different from each other, which is a good thing. We want to have genetic variation, just in case if something happened in the environment and what used to be successful in that environment is no longer successful. If we have genetic variation, then there could be some organisms of that species that have a new genetic variation that might be successful in this new environment. So the species as a whole will survive, right? So that's what we want. We want the species to be able to survive if there is some type of environmental change. Okay, so the result is 4 genetically different haploid cells. In males, we have 4 sperm, and in females, we end up with 1 egg and 3, they're called polar, I always do that, polar bodies. So we do end up with 4 cells, but only one of them actually becomes the egg. Okay, so that was the first page. It says diploid versus haploid. If a regular cell has 52 chromosomes, the somatic cell number would be, so somatic is going to be our like normal cells, like that would go through mitosis. So the normal cell number would still be 52. Cool. So if a regular cell has 52 chromosomes, the somatic cell number would be 52, which is the blank number, a result of mitosis. And that is the diploid number. The gamete number would be, so when we were trying to figure out the gametes, if we had 52 in our original, right, we know that's going to divide in half, and those will divide in half again during meiosis. And we'll end up with each cell half the number. So what's half the number of 52? That would be 26. So that would be 26, which is the haploid, half number, which is the result of meiosis. How many ova/eggs will produce from one cycle of meiosis, going through meiosis 1 and meiosis 2 only once? We'll end up with 1 egg. How many sperms? We would end up with 4 sperm, and we talked about that earlier. How many chromosomes does each human cell start off with at the beginning of meiosis? So we start off with 46 chromosomes in a quote-unquote normal human. How many chromosomes does each human cell end up with after? Wait, what? How many chromosomes does each human cell end off with after meiosis? I think it should say end up with. I don't know, it seems weird. Okay, so how many chromosomes do we end up with after meiosis? So meiosis, we were producing the haploid cells, so that's going to be 23 in each cell. Are these cells identical or genetically different? They are genetically different. What happens between meiosis 1 and meiosis 2? That reduces the number of chromosomes. We skip interphase between the two phases, between the two meiosises. So we go through meiosis 1, So PMAT, and then from telophase 1, we go straight into prophase 2. So we skip interphase, so the DNA is not being replicated, which is what's helping us half the number of chromosomes. Okay, what is it called when homologous chromosomes come together during prophase 1? It's called a tetrad. And when a tetrad is formed, we can then have crossing. Oops, no. Sorry. There we go. Crossing over happen. What phase of meiosis does crossing over take place? Oh snap, we just said that. Prophase 1. What type of cell division ends with haploid cells? Meiosis is the type of division that ends up with haploid cells, right? Mitosis ends up with diploid cells. Okay, so I think that was it for meiosis. I'm going to post this video, and then the next video will be on the review guide for protein synthesis. So you can stay put for that one. Go ahead and pause this.